All right, we are going to go over some uh, information today, just as a general introduction to the scientific method. Even though I'm not here, you still get to learn things because we have technology, so I can film this video. It's great. Um, so in order to jump into really our understanding of biology, we need to talk a little bit about general science because biology is obviously one of sort of the three central branches of science, and it's going to follow the scientific method. A lot of this information is probably going to be review for you. I just need to make sure that we're all on the same page before we move forward, mostly because in this class you're going to be designing a lot of your own scientific inquiries, and you need to know how to do that. And that may not be something you've done in your classes before, so I want to make sure that you're ready to do that. So let's jump in. You've probably all talked about the scientific method or the scientific cycle. It's different, how different teachers think of it, but I really like to think of the scientific method as an ongoing process. So here you can see all the steps are arranged in a circle, and it has this little loop that sort of continues within it until you finally can develop these generalized theories, which is the ultimate goal sort of the scientific method, to develop theories that actually explain what goes on in the natural world. So this whole process is going to start with making observations, thinking of questions, formulating hypotheses, developing testable predictions, and testing those with a procedure called an experiment. At that point, you might realize you weren't right, your hypothesis was not correct, so you go back and you refine, alter, expand, or reject your hypothesis, then you develop more predictions, and you continue forth in that loop until you finally have enough information to actually explain something, and then you make more observations and you start the whole thing over again. So that's why we think of it all as sort of a cycle, which is why we're never done with science. There's always new things to think about and discover, and more often than not, discovering the answer to one question makes you think of three other questions. So we're going to start with our questions, as that is sort of the first step in this whole process. So obviously when we're doing science, we're answering a specific kind of question. These questions need to be measurable and they need to be observable. So examples of measurable, observable questions are like how, what happens when. A good example of what happens when is how does blank affect blank? So how does being tall affect running speed? That would be a good question. Are these two things influence each other, etc.? There's lots of ways that you can see measurable, observable questions. Why questions tend to be more difficult to observe because we can't actually observe the reasons behind something. We can say how something is affecting something else, but we can't always explain exactly why. So why questions tend to be problematic. The other place you get into trouble is when you're asking questions that are very subjective. Like, is this big? Well, what I think is big and what you think are big are two very different things. So we could answer, what size is a thing? But to answer, is it big, is a subjective question. Science doesn't do well with subjective questions. So some examples of questions, right? What is the effect of cigarette smoke on lungs? That is a scientific question. We can measure that. We can observe the effect of cigarette smoke on lungs, and we have. We know that when people smoke cigarettes, their lungs get all black and tarry, and people have trouble breathing, so it affects the function of the lungs. So we've observed that. Why is blue the best color in the world? Well, that's going to be a very subjective question. Not everybody thinks blue is the best color in the world, and it's a why question. So even if blue were the best question in the world, answering why would be very difficult because that's not an observable piece of information. Did the grass turn brown because it needs fertilizer? We could answer that. We could give the brown grass fertilizer, and if it turns green again, fertilizer was what it needed. What is the effect of studying time on test grades? Well, we could test that out. We could do an experiment. We could say, okay, well, this time you're going to test or study for 10 minutes and then take the test. This time you're going to study for 20 minutes and then take the test and see how increasing the time affects the test grade. So that is an observable, measurable thing. Why is basketball a boring sport? That's not a testable question, right? It's a subjective question. Not everyone thinks basketball is boring. Many people think basketball is exciting because, it kind of is. But it's also a why question. And those whys, like we said before, are very difficult to answer because why, the reasons aren't observable. They're not measurable. We can't observe that. So there are whole other realms of study to answering why questions. That's not what science really does. So then the next step, once you've developed a good scientific question, is to develop a hypothesis. A hypothesis is like a predicted answer. It's a prediction. It's what you think is the answer to your question. And it's educated. That's going to be based on research and background knowledge. It's not just like, oh, well, I think this is true. But this is the point in the experimental method where scientists would be reading what other scientists have done in this area and really looking up what do we already know about this and what do I reasonably think is going to happen. The hypothesis also needs to be testable. It needs to be something you can test out. And it needs to be measurable, just like your question. Usually we will write a hypothesis as if blank, then blank, because blank. The because part brings in your research, the reasoning. It's what makes sure that this is educated. The if and then refer to like if this happens, then that happens. So like if we add fertilizer to our brown grass, then it will turn green because fertilizer helps grass grow, right? So like that is an example of a hypothesis for a relatively straightforward experiment. So again, constructing your hypothesis, you want it to be logical, you want it to be based in fact. If I feed my plant energy drink, it will grow faster. That example is missing a because energy drinks provide chemicals that organisms need to move quickly. Right? That would finish out that hypothesis. So remember, your hypothesis should always explain what you expect to happen, be clear and understandable, be testable, measurable, and contain two of your variables. Speaking of variables, once we have our question and our hypothesis, we're going to develop an experiment. The experiment is your procedure that you're going to use to test your hypothesis, to see if your hypothesis is correct. And you can accept your hypothesis as is if your hypothesis turns out to be supported by your experimental data. You can reject your hypothesis if it turns out to be proven wrong by your data, or you can alter your hypothesis, change it a little bit and go back and redo an experiment to see if you get a better understanding. Designing an experiment uses what we call variables. Variables are anything that can change from one trial to the next in an experiment. And so variables can be classified really into independent, dependent, and constants. The control isn't really a variable. Sometimes you will see things referred to as control variables, and your previous teachers may have used the term control variables, but the control is actually a group. It's one of your trials that you do in your experiment. It's the group that you're comparing everything else to. So it's your baseline for comparison. We'll go over some examples in a minute, but it's basically the experimental trial where you're not changing anything. It's what happened naturally, and that's going to give you your baseline so that you can tell if anything changes in your other trials. The term we're going to use for variables that we don't change from experiment to experiment is constants. These are things that we hold the same. So a constant is a variable that does not change throughout the entire experiment in any of the trials. And it remains the same. These are the things you're not trying to test. You're trying to keep them all constant. Now, the other two variables that you are trying to change fall into two forms, independent variables or dependent variables. Independent variable is the variable that you're changing. It's the if in your hypothesis. If the independent variable changes, then your dependent variable would change. Your dependent variable is your result. It's your outcome. It's what you think is going to happen because of your independent variable. So the independent variable is the cause, and the dependent variable is the effect or the outcome. Sometimes the independent variable is called the manipulated variable. Sometimes the dependent variable is called the responding variable because it's responding to the manipulated variable. So independent and dependent, sometimes called manipulated and responding. These other factors, these confounding variables, that's what we're trying to avoid with the use of constants. We're trying to keep everything else the same. All these other factors we want to keep constant so that our experiment doesn't have confounding. Confounding means like confusing variables. If every other factor is exactly the same, then we can reasonably conclude in our experiment that the effect, the dependent variable, any change in the dependent variable, was actually caused by our independent variable. And that's what we want. We want experiments that can actually draw conclusions, useful conclusions about the world. Excuse me again. So we're going to set up a control group which is going to be our basis for comparison. That's normally the sort of typical situation where we haven't changed anything. Then we're going to change the independent variable from one trial to the next. We're going to measure the dependent variable from one trial to the next, and we're going to keep everything else constant, all the same. So here's an example of an experiment. So in this experiment, the question is sort of, does the moisture content of the environment affect the activity of these organisms? So we have two containers here that are connected by a little doorway in each of our trials. In one of our trials, our experimental trial, one of the containers is wet and the other container is dry. So they have different moisture levels. That's our experimental trial. In our control trial, the baseline that we're going to compare our results to, both sides are wet. So we're going with the default position of there not being a dry environment, that everything is wet, so they're the same there. But then there are other aspects of the environment that might affect these organisms, right? Light, the kind of filter paper you're using, the temperature, the number of bugs that you start with, those are all going to be possible confounding variables. So in our experiments, we're going to keep all of those factors the same, right? We're going to use a 25-watt light bulb for all of them. We're going to use a particular brand of filter paper of a particular size, so the #1 Wattman brand, and we're going to keep it the same for everything else. We're going to keep all of them at 20 degrees Celsius. We're going to put 5 bugs in each container at the start, and then we're going to observe the movement of those bugs. So our dependent variable would be the populations of bugs at the end. If all of these dry bugs move into this wet environment, we could reasonably conclude that the bugs prefer a wet environment. That if the control group moved relatively equally, then we would have a different motion in this side than in this side, and then because we kept everything else the same, we could reasonably conclude that that was because of our independent variable, because of our change. If they all ended up in the dry side and these guys all stayed relatively evenly spread out, then we could reasonably conclude that the bugs prefer a dry environment. And so that's the whole point of running an experiment, to try to be able to draw reasonable conclusions. So I hope that example helps to clarify the difference between control, which is our experimental group that's serving as our baseline comparison, and constants, which are variables that could confound our experiment, so we keep them the same throughout all of the trials. In a perfect world, you would have one independent variable, one dependent variable, and everything else would be exactly the same. That's the goal in our perfect experiments. Everything else would be a control variable. Everything that could possibly be kept the same should be kept the same except the independent variable. That way you know that whatever happens to the dependent variable happened because of the independent variable. And so that is how we're going to set up our experiments when we do them in class. We're going to pick an independent variable, pick a dependent variable by developing good testable questions and good testable hypotheses. Then we're going to design a procedure that allows us to test that by keeping lots of things the same, setting up a control group, a basis for comparison, and then experimental trials that change the independent variable but keep everything else the same. And that's how scientists work. Once they finish up with that gathering of data and testing their predictions, it can go a number of ways. You can go refine, alter, reject your hypothesis if it didn't come out to support your hypothesis. Then you go back, you develop different predictions, you test them again, and you keep doing that until you can move into the last step, which is developing a general theory, like the theory of gravitation or like the theory of evolution. Scientists use theory differently than the general world uses theory. Theory in science means it's a hypothesis, a prediction that has been upheld or has been proven seemingly true, because we can't prove anything to be absolutely true, but proving seemingly true through many, many, many experiments. So in the case of like the theory of evolution, it's been hundreds of years of experiments that have upheld the hypothesis of evolution. Every experiment that we do adds to our understanding of evolution, but upholds that theory of natural selection. So that is sort of your general overview of the scientific method. I hope that was helpful. Now you have a practice worksheet to get some practice using those variable terms, and we'll go over that class tomorrow. Have a great day.